morning, and today I'm very happy to introduce the final defense of Varsha. So Varsha came to my lab in fall 2009, and she came just at the same time as I started the lab. And Varsha, she has always been very helpful. She has been excessively patient with me, and I appreciate this. And she has produced a lot of work. So today she's going to present only a part of this work, and I will let Varsha talk now.
Thank you, Dr. Dekler, for such a nice introduction. I don't think I could have done all this without your help. Thank you very much for everything. So, good morning all of you, and welcome to my final defense. Title of this presentation is Receptor for Advanced Glycation and Products, that is RAGE, in melanoma progression. In this study, we have determined role of RAGE in melanoma progression, and we have also determined the downstream signaling involved. So before going into the detail of this presentation, I would like to talk about melanoma and how RAGE could be a potential therapeutic target in melanoma. Melanoma is a type of skin cancer originated from melanin pigment producing cells, melanocytes. It is the deadliest form of skin cancer because of its high metastatic rate. Patients with primary melanoma tumor have more than 90% 5-year survival rate, which drops to less than 20% in case of metastatic melanoma patient. In primary stage, melanoma tumor cells have more proliferatory phenotype and less metastatic phenotype. Therefore, these cells tend to proliferate to form a tumor at the site of origin. Once these tumor grows to a sufficient size, melanoma cells switch from a proliferatory phenotype to a metastatic phenotype, and these cells migrate to another part of the body. Once they reach to another site of the body, these cells switch back from a metastatic phenotype to a proliferative phenotype in order to generate another tumor at that site. This switching process of melanoma leads to heterogeneity in melanoma cells, and therefore it makes melanoma difficult to treat. It is very important to understand the mechanism of melanoma progression in order to develop better therapies against melanoma. Because currently there are very few treatment options available for melanoma. Up to year 2011, dacarbazine was the only treatment option available for melanoma patients. It is a chemotherapeutic drug of alkylating agent category. It alkylates DNA and therefore inhibits DNA replication, and therefore it kills cancer cells. Like other chemotherapeutic drugs, dacarbazine also have lots of non-specific side effects. Due to urgent need of better melanoma therapies, FDA has approved 2 new anti-melanoma drugs in year 2011. One is ipilimumab, which is a monoclonal antibody against cytotoxic T lymphocyte antigen 4. Another is vemurafenib, which is a BRAF inhibitor. Although emergence of these new drugs have improved the survival of melanoma patients, however, these new drugs are also associated with many side effects and other limitations. Therefore, there is still a need to identify new target and to develop new therapies against melanoma. Here, we have focused RAGE as a potential therapeutic target in melanoma. RAGE is a cell surface receptor of immunoglobulin superfamily. As you can see in this image, it has one extracellular domain which is responsible for ligand binding. It has a small transmembrane domain which holds RAGE with the cell membrane, and it has a cytosolic domain which is important for signal transduction. Although there are many more RAGE ligands identified, but here I have just shown 3 RAGE ligands: SN-RAGE proteins, HMGB1 proteins, and advanced glycation products, because these RAGE ligands have been shown to be associated with melanoma. Since RAGE has this ability to interact with various structurally different ligands, therefore RAGE is known as pattern recognition receptor. Upon ligand binding, RAGE leads to activation of multiple downstream signaling pathways, including MAP kinase, JNK, AKT, which in turn leads to activation of transcription factor and FKB, which regulates transcription of various cancer promoting genes and therefore leads to cancer progression. Extracellular domain of RAGE without transmembrane splicing process of pre-mRNA during the maturation process of mRNA. The soluble RAGE is also known as decoy receptor, as it can interact with RAGE ligand, but it cannot initiate any downstream signaling pathways. Therefore, soluble RAGE can inhibit RAGE-mediated signaling by inhibiting the availability of RAGE ligands to cell surface express RAGE. The mechanism of RAGE-mediated signal activation is very complex and is not very well understood. Few reports have shown that RAGE molecules tend to dimerize or oligomerize on cell surface, and this oligomerization process is important in signal activation. As you can see here, this RAGE is dimerized on the cell surface and leads to activation of signal. Upon ligand binding, the oligomerization process increases and leads to amplification of the signal. RAGE and its various ligands have been shown to be associated with many cancer types, including melanoma. Inside the tumor, RAGE is not only expressed on cancer cells, but also expressed in surrounding stromal cells, blood vessel, endothelial cells. So RAGE, together with its ligands, create a pro-inflammatory tumorogenic microenvironment inside the tumor by increasing expression of certain cytokines and growth factors. And it has been also shown that accumulation of RAGE ligands itself can lead to upregulation of RAGE. So via this feedback mechanism, RAGE and its ligands can lead to amplification and prolongation of tumorogenic signal inside the tumor, which leads to further increase in cancer growth. In the next slide, I will be talking about two of the RAGE ligands, advanced glycation end product and assembled proteins. Advanced glycation end products are first known ligands of RAGE. Therefore, RAGE got this name, receptor for advanced glycation end products. These are non-enzymatic adducts of proteins with sugar. They can be generated inside the body, or they can also be generated outside the body during certain cooking processes. And this advanced glycation end products have many pathogenic roles to play inside the body. A group of researchers have shown that advanced glycation end product as well as receptor for advanced glycation end product is expressed in human melanoma tumor. They have also shown that melanoma cells upon treatment with advanced glycation end product leads to enhanced proliferation and enhanced migration, which implicates an important role of age and RAGE in melanoma progression. Acylate proteins are another group of RAGE ligands. These are calcium binding proteins, and acylate B is one of the member of acylate protein superfamily. Acylate B is the best characterized ligand of RAGE. Acylate B is also expressed in melanoma cells. Inside the melanoma cell, acylate B interacts with tumor suppressor p53 protein and inhibits its tumor suppressive activities, and therefore leads to enhanced melanoma progression. Acylate B has also shown to inhibit expression of p53 in melanoma cells. Acylate B is also released from melanoma cells as well as melanoma tumors, and its serum concentration correlates with disease progression. Therefore, serum concentration of acylate B is clinically used as a prognostic biomarker for melanoma. There are few reports that have shown association of RAGE in melanoma. In year 2003, a group of researchers have performed a tissue micro study on melanoma tumors, and they have found that 10% of tumor tested were strongly positive for RAGE. In subsequent year, another group of researchers have shown that both age as well as RAGE are expressed in melanoma tumor, as I have shown in my previous slide, and they have also shown that melanoma tumor growth can be suppressed upon treatment with anti-RAGE antibody in xenograft mouse model. In year 2009, Dr. Leckler and Dr. Vetter have performed another study in that they have assessed mRNA expression level of RAGE in different melanoma patients, and they have found that RAGE mRNA expression was highly variable between patient to patient, and they found 50-fold difference in RAGE mRNA expression between different patients. And they have also found that RAGE mRNA expression was significantly higher in stage 4 melanoma patients as compared to stage 3 melanoma patients. So based on these reports, we hypothesized that RAGE, when overexpressed, plays a role in melanoma progression. So to prove our hypothesis, we have designed our research strategy. We selected 2 melanoma cell lines which were originated from a same melanoma patient. WM115, which was obtained from a primary melanoma tumor, and WM266, which was obtained from a metastatic melanoma tumor. In the next step, we have stably transpected WM115 cells with full length RAGE in order to generate RAGE overexpressing melanoma cell line models, and we obtained 2 clones of RAGE overexpressing cells with 2 different expression level of RAGE. We simultaneously transpected both WM115 and WM266 cells with non-coding antiplasmid in order to generate WM115 mock and WM266 mock cells as control. The goal of this study was to assess effect of RAGE overexpression on WM115 primary melanoma cell line and compare it to that of WM266 metastatic melanoma cell line. After generation of these cell lines, we have characterized these cells for the presence of RAGE at both mRNA and protein level. mRNA expression level of RAGE in these transfected cells was assessed by PCR, and amplified product was run on agarose gel. And as you can see that WM115 RAGE cells have higher mRNA expression level of RAGE as compared to WM115 mock. We next quantified RAGE protein in transfected cells by ELISA, and our result showed 11 picogram, 81 picogram and 1000 picogram of RAGE per mg of total protein in WM115 mock, WM115 RAGE 10x and WM115 RAGE 100x cell lysate. In WM266 mock, we found 24 picogram of RAGE per mg of total protein. We have also quantified soluble RAGE protein in the cell supernatant of transfected WM115 cells, and we found 400 picogram of soluble RAGE per ml of cell supernatant in WM115 RAGE cells, whereas in WM115 mock cells, soluble RAGE level was below detection limit. We next determine whether RAGE is expressed on the cell surface or not. So for that, we have performed a flow cytometric titration on mock and RAGE transfected WM115 cells with increasing concentration of anti-RAGE antibody. And as you can see, mean fluorescence increases with increasing concentration of antibody in WM115 RAGE cells, whereas in mock cells, mean fluorescence remain in the baseline level and remain constant. Next, to visualize RAGE on the cell surface, we have performed immunofluorescence, and as you can see, we detected RAGE on the cell surface of WM115 RAGE cells, whereas in WM115 mock cells, RAGE expression was very low and we couldn't detect in our experimental conditions. So these data show that RAGE is not only expressed in the cells but is also present on the cell surface and is available for ligands to bind.